Hi there, my name is Courtney Richardson and I'm going to be doing my project. So let me share my screen. Okay, and let's get started. There we go. Okay, let's get started. Okay, so RSV is a virus that reaches the lower respiratory system that can overcome many different defenses of the host's immune system. I'm going to do my presentation today on RSV, or respiratory syncytial virus. This virus is very common during the winter and spring months, and it can be dangerous to older adults, those with compromised immune systems, or babies and young children. This common respiratory virus affects the lungs and smaller airways called the bronchioles. It is estimated that the majority of children will contract this virus before they are of 2 years of age, and only a small percentage results in the need for aggressive treatment or hospitalization. Typically, the only treatment or care focused on comfort and is needed to combat the symptoms. Respiratory syncytial virus is highly contagious, especially during the first few days up to a week after a person first demonstrates symptoms. Those with compromised or weakened immune systems could even remain contagious for up to a month. While this virus is spread through close contact, the main method of transmission is through droplets from sneezes or coughs. Droplets can also infect a person if they are on a surface that the person touches, typically, and then puts into their mouth. And typically, symptoms take a little while to develop and show up. According to the CDC, the main symptoms of respiratory syncytial virus are similar to a flu or a cold. However, in most serious cases, inflammation of the bronchioles and pneumonia can occur, especially in very young children or much older adults. Every year in the United States, respiratory syncytial virus leads to roughly 2.1 million outpatient visits for children under 5 years old, 58,000 hospitalizations among children younger than 5 years old, and almost 200,000 hospitalizations among older adults categorized as above 65 years, and roughly 14,000 deaths among older adults. The most popular season for RSV is during the colder months, particularly fall, winter, and spring. The U.S. Department of Health and Human Services reported that with RSV infection, the season peaked through late December to mid-February on average. Laboratory detections within the United States use the National Respiratory and Enteric Virus Surveillance System. This system is a voluntary and laboratory-based method of monitoring trends in several different viruses, and they track the number of RSV tests that are positive. It sorts them according to specimen type, date of collection, and collection location. This data is essential to public health officials in tackling the challenge of reducing RSV infections. Respiratory syncytial virus is similar to coronavirus strain COVID-19 because they are both types of respiratory viruses, and the symptoms are very similar. However, with COVID-19, symptoms in children are typically mild, such as a small cough, a small runny nose, or a little fever. However, adults seem to have more severe symptoms, which can even include trouble breathing. And with RSV, children have more severe symptoms than adults, so it's kind of switched, which can lead to a similar effect on their breathing problems. Additionally, having RSV could lower an individual's immunity, which can increase the risk of contracting COVID-19 for both children and adults. RSV is an increased risk for infants that were preterm younger than 6 months, children who have congenital heart disease, chronic lung conditions, and children that have neuromuscular diseases such as muscular dystrophy. Repeat infections are very possible, especially in or even in close proximity to previous infection. But thankfully, reinfection, especially during the same season of RSV, is typically less severe. In some cases, a middle ear infection occurs, which is primarily in babies and young children. And now I wanted to talk about a study performed by the American Association of Pediatrics, whose objective was to evaluate the safety and efficacy of treatments to prevent severe RSV in infants born prematurely. Data from a randomized blind multi-center trial during 3 consecutive RSV seasons was analyzed. There were 249 children involved in the study, with 162 of them being premature. In addition, 102 of these children had bronchial pulmonary dysplasia, and 87 children involved in the study had congenital heart disease, so they were excluded from this study. The children involved were randomized to receive monthly injections of different dosages of the respiratory syncytial virus immunoglobulin. Their results were from premature infants with and without bronchopulmonary dysplasia were examined and compared to the control group of infants who did not receive this injection. It was found that children with the high dosage of respiratory syncytial virus immunoglobulin had a significant reduction in the instances of RSV infection. Additionally, there was a moderate to severe decline in RSV-associated hospitalizations, as well as a decline in admittance to the intensive care unit in these cases. However, unfortunately, there was adverse reactions in roughly 5% of the children that received the RSV immunoglobulin injection. The study concluded that prophylaxis with the injection is currently the only effective method of attempting to prevent RSV infections, especially in some severe cases that require hospitalization and admittance to the ICU in high-risk preterm infants. RSV is commonly misdiagnosed as the influenza virus, which is part of the reason it is so dangerous. The name respiratory syncytial virus is derived from its ability to cause cell fusion when grown in cell culture. Several of the virus cells can fuse together to form a very large multinucleate cell, which is known as a syncytium. These large cells are commonly produced from infections by viruses such as RSV. One is able to be infected by RSV multiple times in one season due to a poor naturally acquired immunity. There has been an immunoglobulin product approved that potentially can protect infants that are immunocompromised or have lung problems and are at a high risk for severe complications if they become infected with RSV. The only pharmacological therapy available for the treatment of RSV is a nucleoside analog ribavirin. This drug can be administered orally or by inhalation. Ribavirin must also be activated through the process of phosphorylation to be effective. The prevention of RSV is still being studied and in clinical cases, which we will talk about further in a moment. However, a humanized mononuclear antibody is possible to be injected as a form of prevention when it is acted with RSV. Non-specific antiviral proteins known as interferons are produced by host cells as a response. The production of interferons leads to a production of protein that prevents the synthesis of some viral components from nucleic acid. Interferons have a broad spectrum antiviral activity and inhibit the growth of cancer tissues as well, as a bonus. And here's the ribavirin. An RSV infection of children who were previously immunized with a formula and inactivated vaccine is associated with exaggerated memory of the Th2 response. This potentially could cause an enhanced disease and pulmonary eosinophilia. This is consequential due to the adverse effects of vaccination testing, and the development of a safe vaccine against RSV is still being tested. A critical first step in the development of a safe vaccine is to study in detail the prevention of vaccine-associated diseases such as pulmonary and eosinophilia. Thankfully, the mouse model of RSV infection mimics the human model closely, and mice are commonly used to test immunizations. Mice have been previously immunized with a recombinant vaccinia virus that expresses the attachment of a glycoprotein, which exhibits extensive lung inflammation and other complications such as pulmonary eosinophilia and enhanced disease. Th2 cytokine cells take part in the response enhanced diseases following an RSV infection due to their ability to eliminate eosinophilia when they are depleted. Recent studies have demonstrated that CD8 T cells, which are RSV specific, can also inhibit Th2-mediated pulmonary eosinophilia. The mechanisms for this are unknown, and information gained from animal testing will additionally provide information that will potentially lead to the design of an effective and a safe vaccine against RSV. The respiratory syncytial virus is considered to be a medium-sized RNA-enveloped peroxymyovirus of the genus pneumonovirus. The genome consists of roughly 15,000 nucleotides, which encodes 10 vital proteins. The nucleocapsid and protein tightly encapsulated the genome, which in combination with the protein P and large subunit L is able to form the minimum unit for an RNA replication. RNA replication of RSV involves an exact copy encapsulated intermediate known as the antigenome, which serves as a template. Two non-structural proteins known as NS1 and NS2 have unknown functions, but it is hypothesized that NS1 could potentially be a negative regulatory factor for RSV synthesis. The G protein of RSV is heavily glycosylated and composed of roughly 300 amino acids. Variability in the G protein accounts for the main differences in the most common RSV strains, and this is also how the cells are able to recognize microbial products through the pattern recognition receptors. CD14 and TLR receptors are the primary response for both gram-positive and gram-negative bacteria. When a patient is infected with RSV, the most important component of the immune system response are the phagocytic cells, or neutrophils, macrophages, specifically eosinophils and natural killer cells, which can also play a role in the battle against RSV. A recent study of the RSV infection in infants found that 93% of cells in the upper airway were neutrophils, suggesting that neutrophils play a very important role in the pathological changes that are involved in RSV bronchiolitis. Respiratory epithelial cells and macrophages produce a chemokine IL-8, which promotes the production of neutrophils chemotaxis. Gene expression, which shows a pattern of IL-8 in the respiratory epithelium cells exposed to RSV, have also been observed in vitro studies. In addition, macrophages are essential in controlling the immune response to a viral infection, such as RSV, through the production of cytokines as well as direct interaction with T cells. Since macrophages are the first cells to encounter a potential RSV infection within the airways, they are responsible for the primary stimulation of the immune system to respond to the potential invasion. Apoptosis of mononuclear RSV-infected neonatal cells were observed to decrease more rapidly than in adult cells, which potentially could explain the mechanisms which cause a more severe response in young children, as opposed to non-immunocompromised adults. The series of studies concluded that RSV is the most prominent cause of lower respiratory tract infections in infants and can cause a wide range of other clinical diseases in infants, with the most severe being respiratory failure. Inflammatory changes of the immune system within the lung can cause clinical symptoms of bronchiolitis, and additionally, the clarification of pathogenic mechanisms involved in RSV could potentially provide an explanation for long-term consequences of RSV, such as asthma. An increase in asthma for recurrent wheeze and clinical allergies up to the preteen years have been reported in past hospitalization cases of RSV. The potential link to this was studied to see if this was just a fluke or whether the connection was within the airway functions in RSV infections and asthma. This study followed the children up to age 18 that had RSV in the past, and this included clinical examinations, blood tests to measure eosinophils, skin prick tests, antibody tests, and questionnaires to assess the prevalence of asthma, clinical allergies, and perennial allergies within individuals who had RSV as children. Persistent wheezing was associated with early allergic sensations found in RSV cases, which is roughly 30% compared to control groups of 1%. It was also concluded that severe RSV bronchiolitis is associated with allergic asthma persisting into early adulthood, which may reflect airway remodeling after an RSV infection. This figure shows the proportion of current asthma or reoccurring wheezing, current allergic rhinitis conjunctivitis, and current positive skin prick test to find perennial allergies in respiratory syncytial virus, in addition to the control groups, as well as taking into account hereditary asthma. I wanted to take a minute to discuss the trends and hospitalization rates and risk factors for cases of severe bronchiolitis that have occurred after RSV infections. A study by the American Academy of Pediatrics was performed to determine the burden of hospitalizations related to RSV infections within a children's hospital, as well as to identify the potential risk factors associated with severe disease. Medical records of patients hospitalized for bronchiolitis were examined through clinical microbiology and demographic characteristics, including the presence of potential underlying medical conditions that may affect this study. Children with RSV and non-RSV bronchiolitis were evaluated for differences and their risk factors for severe disease. Within the patients younger than 2 years of age with bronchiolitis that had been hospitalized, it was examined that RSV infections can cause a significant increase within a 5-year study, which was done from 2002 to 2007. Patients that had RSV bronchiolitis were typically younger at their hospitalization and had a lower percentage of potential underlying medical conditions that could affect their ability to fight the virus. Disease severity defined by the length of hospitalization and the requirement of supplemental oxygen, as well as the intensive care unit admittance, was significantly more severe in children that did have RSV bronchiolitis. It was concluded that there was significant increase in hospitalizations for bronchiolitis that resulted from RSV within the 5 years as well. Majority of children that had RSV bronchiolitis were previously considered to be generally healthy. However, the disease severity was far worse compared with children hospitalized with non-RSV bronchiolitis. So this concludes my research on respiratory syncytial virus as related to potential severe disease and bronchiolitis, taking into account studies performed by the various researchers mentioned in the presentation, especially during RSV season, which we are in RSV season currently. It can be extremely dangerous for new babies to be exposed to large numbers of people, which puts them at risk for contracting RSV, especially since adults do not have as severe symptoms. So it may seem like a flu or a small cold. It could potentially be very harmful for an infant, especially within the recent coronavirus pandemic. It is becoming increasingly popular to limit contact with new babies. And especially after my research on this topic, I am in full support of limiting contact with people for new babies according to how the parents of the child see fit. So, thank you for listening to my presentation.